Hey everybody, this is Mrs. Mancuso, Merm here. Today we're going to talk about regulation of genes. So to get started, let's think about this. All of your cells have the exact same genome, meaning they all have the same DNA, but only some of those genes get expressed in each cell. So for example, you have the DNA to make enzymes that break down the food that you eat. And that DNA that has the gene to produce the enzymes is in all of your cells, but it actually only gets expressed in areas like your stomach and your small intestine because that's where digestion occurs. Other cells, like muscle cells, have the gene to make that enzyme, but they do not express it. So this video is all about how that occurs. So control. This is at the transcriptional level, which means you want to control if a segment of DNA is made into RNA or not. So the process of transcription is making RNA using DNA as a template. You want the control at that level because that's right at the beginning. It's before the protein is made. All right, so there's a difference between what prokaryotes do and eukaryotes do. So we're going to start with prokaryotes, our bacteria. They have these units called operons. So the parts of an operon are really important to understand. So first of all, it's a segment of a gene. All right, within it, there's a regulatory gene that produces repressor proteins. And I'm going to show you a visual in a minute so you can really put this all together. A promoter, which you already know that, the promoter is the area where the RNA polymerase is going to bind to the DNA. Then there's an operator, that's like a little on-off switch. That's where the repressor protein binds. And then, of course, you have structural genes. So structural genes are the genes that have to get transcribed in order for you to make a protein. All right, so here is our picture. This is a Lac Operon that we're going to use for an example. And first, I'm just showing you what everything is. So this black strand right here, that's the actual DNA. So if I could look up close, it would be all letters like this TATA. It would be letters the whole way across. A's, T's, C's, and G's. The beginning portion of it, so upstream, is the regulatory gene. The regulatory gene makes repressor proteins. So when I say it makes repressor proteins, it codes for messenger RNA that goes to the ribosome and builds this repressor protein. All right, the promoter is where my RNA polymerase binds to, right there at the TATA box. The operator is the on-off switch. This is where the repressor protein can bind. The structural genes are the genes that make messenger RNA. So this is the thing that we actually care the most about, these structural genes. They make the RNA to make the proteins. So in our example, this is going to be the protein they make, lactase, and this is the job they do. They got to break apart lactose. All right, so we're going to get started with our Lac Operon here. So the black is our DNA. This red is the repressor protein that was made according to this regulatory gene, and it's right here. So right now, there is no transcription happening. This cannot move because the repressor protein is in the way. So in order to make it start working, we need this. This is lactose. Lactose is a disaccharide, which is why I drew it like this. So when this is present, my repressor protein has a stronger attraction to the lactose than it does to the DNA. So it moves off of the DNA. This allows my RNA polymerase to now move forward. So as this moves forward, it starts coding. Like I'm going to need a C and a G and an A and an A and a U and a C and a G and an A and a U. And it builds this messenger RNA. When it builds the messenger RNA, that will leave the nucleus after it gets its cap, its tail, and its splicing, and it'll go to the ribosome and it will do translation and it will build these proteins. So these proteins are the lactase. So these proteins are going to get to work on the presence of this lactose. So when they come in here, they will break the lactose into monosaccharides. And it'll come over here and it'll break this one, breaks it into monosaccharides. And it'll come over here and it'll break this one. Now, when this is broken, now the repressor protein has nothing to attach to anymore. It's not attracted to the monosaccharide. It was attracted to the disaccharide. So it's going to go back up here. Actually, my RNA polymerase made it all the way over here because it was able to transcribe this section. So now it is up here again, which means when this wants to start working again, it's now blocked. So when the repressor protein is at the operator, transcription is off. When the disaccharide lactose is present, this repressor protein moves out of the way, and now transcription is not blocked and it's on. So that's why we call it an inducible operon. It could be induced to turn on. Now we're going to talk about the other kind of operon. This is repressible. So repressible means it's usually on. It is usually doing transcription, which means it's making proteins. So this is something that you don't need, like, hey, there's no milk present, so it's not on. Nope, this one's on all the time. But since it is repressible, that means you could turn it off. When would you want to turn it off? If you made too much stuff. So if you make too much product, you got to stop. You got to turn it off. You don't need all of that. So a good example of this is the trip operon. Trip stands for tryptophan. It is only turned off when too much tryptophan is made. Why make more if you don't need it? So let me show you what that looks like. Here is my DNA. Here is my repressor protein, but the repressor protein right now is inactive, which means it can't bind here. So what happens? Transcription is on. The RNA polymerase goes to the operator, goes to the structural gene, makes this RNA, goes to the ribosome, makes this tryptophan. This tryptophan is attracted to the repressor protein, and they together make an active repressor protein. As an active repressor protein, now they block the way. So it was on, now it's off because I got too much of it. So when it's off, now when this goes to work again, it's blocked and it can't get any further. So that's how a repressible protein works. And eventually, as this gets used and this gets used and this gets used by your body, this one is going to need to be used. And when it's used, this is now inactive and goes off the space. And now this can transcribe again and make more tryptophan. So that's how that one works. All right, but in us, in humans, in eukaryotic cells, we do not use operons. We also do not have circular DNA. We have linear DNA. Remember, a bacteria has circular DNA. We use what are called transcription factors. Transcription factors are proteins. They're very specific. They each have their own shape, and they bind to the promoter, and they really do the job of calling in, like, hey, RNA polymerase, get over here. We need you to work. So the RNA polymerase attaches and does transcription wherever the transcription factor is. So what makes a transcription factor? A transcription factor is made from a signal transduction pathways. So remember when we learned about that earlier this year, you needed the ligand, and the ligand was received by a receptor protein, and it started that phosphorylation cascade, and it resulted in transduction and then eventually a response. Well, the response was the protein. The protein was the transcription factor. So it has to be signaled, which is a great level of control in itself. All right. This activity, the activity of a transcription factor, it can be enhanced. You have activators, literally things like broccoli in your food and garlic. Actually, broccoli is more of a methylator. Garlic is more of an activator. So it activates, it makes transcription more likely. Literally things that you eat can change the expression of your genes. All right, they can also be hindered. This is the broccoli one. Broccoli is known to be a methylator. Methylation is the silencing of genes. So when you silence a gene, what happens is you give it a methyl group. We call this understanding epigenetics. So the understanding of how are genes expressed, what factors can influence them. And if you just think for a minute, obviously your food has a huge role in how your DNA is expressed. That's why we say it's so important to like eat well and eat food that is naturally grown, doesn't have a lot of preservatives, because each chemical probably plays a role in affecting how your DNA gets expressed. So this whole science, this whole understanding of how your genes get expressed, that's called epigenetics. The word epigenetics literally means above the genome. So not the actual letters, not the letters don't change, but what does change are these tags and codes that are above the DNA. So over time, your DNA changes, not the letters, but the overall, like how its shape is, even how the ends are, the coiling of the DNA and tags on the DNA. That is really the study of epigenetics. So one of the very, very key words of epigenetics is the word methylation. So way back in biochemistry, you learned that a methyl is CH3. There is nothing polar about CH3. It's a nonpolar molecule. When you add a nonpolar molecule to something, it silences it because it is very hydrophobic. So it tends to hide the gene. If you put a methyl tag on DNA, you're going to hide it. You're going to make it not likely to be expressed. So there's two ways this can happen to your DNA. It could be a straight up tag. Put a methyl tag on DNA and it won't get expressed. Or the other way is to methylate the histones. The histones, remember, are the proteins that DNA coils around. Coil it tightly and it's not very likely to get expressed. All right, this I already alluded to. The things that are the factors are diet, exercise, chemicals, medication, stress. All of those things can affect methylation and the expression of genes. And then the last little note I have here, isn't this just so interesting and cool to understand? What if we could silence genes? Like, what if you know you have BRCA, the breast cancer gene? Well, you don't want to express it if you have it. If there's a way to methylate it, wouldn't that be so great? And if you had other genes, like this makes you have a high metabolism and this makes you have a really sunny disposition, like maybe extra certain brain chemicals that make you happier, wouldn't you want to express those? So super interesting field of genetics that we are discussing now. All right, see you soon, guys.